Our next speaker is Dr. Yulong Li, also from Peking University. And I'll give him just a moment to set up. He will be talking about spying nerve communication by constructing new genetically encoded fluorescent probes.
Well, it's great to be here to see some old friends and meeting new people. And I really appreciate the organizer. In fact, one of my friends commented the design of this tag, and he said it really looks like I'm a new groom because of this tag. So I think we should carry this tradition back to China. So for our speakers, we should have this really nice design. But she also commented I'm getting fatter and fatter. So I think the Chinese food there is really good. So that will be a big reason for a lot of colleagues in the States to visit. So. Today, my talk is about spying nerve communication by constructing new genetically encoded fluorescent probes. As we know, and for Dr. Zhou Zhuan's nice introduction, the brain are very complicated material composed of many different type of neurons, and he just talked about the dopamine-ergic neurons and acetylcholine release, how the different interplays between these two systems. And to execute these higher brain functions, and we of course have the single neurons, but they form microcircuitries, and they connect to regulate different aspects of higher brain functions. So one way to study the brain function is, as Dr. Zhou Zhuan said, we can use good electrophysiology, which give us superior temporal resolution to detect in millisecond resolution how the transmitter release and where the action potential occurs and how it propagates. But the complex anatomy actually prevent most of the electrophysiologists to study many neurons at the same time. And one way to solve this problem is by using optical technique. In fact, more than 10 years ago, when Gary Messenberg in Dr. James Rossman's lab, he designed a fluorescent sensor that is pH sensitive. When this pH sensitive fluorescent protein gets tagged into the synaptic vesicle, which is acidic, upon exocytosis, this acidic environment will meet with the neutral environment, which is outside of the cell, and this correctly targeted fluorescent probe will become neutral, and therefore report the fluorescent changes and reflect the transmitter release. But after these 10 years, we are still using the green ones. There's no many other color available. So when I was a postdoc in Dick Chen's lab at Stanford, I decided to tackle this problem by creating more colorful combination of probes. The idea is very simple. If we can generate many different color combinations, then we can use in genetic ways to put them into different sets of neurons. And the combination of the color and simultaneous imaging will allow us to visualize the synapse transmission in parallel, and therefore hopefully can help us to decode the complex of the neuronal circuitry. So through molecular genetics evolution, starting with M. Strawberry, developed by my postdoctoral advisor's brother, Roger Chen's lab, and we introduce mutations into the fluorescent probe and then generate a pHTomato, mainly because of their Chen family, or like fruits, the tomato, and because it's very pH sensitive, and the spectra is similar as the Roger's TD tomato in terms of the color spectrum. As you can see that in the neutral pH, this pHTomato fluorescent protein is as bright as the well-used EGFP. But the property is that it is different colored and is very pH sensitive. As you can see that by changing the pH from low to high, that the fluorescent intensity increase not only in E. coli but also in mammalian cell. Of course, using this method, then we can have two colors to allow us simultaneously imaging different neurons. And this is a comparison with the green pHTomato target to the synaptic vesicle, and the red one and the green one, they perform similarly. And because of the color separation, then we can imaging the cell signal transduction. If in the same cell, for example, in this case, we could use the genetically encoded calcium indicator, GCAN3, which reports the calcium signal. And as you can see, if you stimulate the neurons, their intracellular calcium will rise due to the activation of voltage-gated calcium channels, not only in the soma, which is shown here, but also in the neurites. But if you look at the pHTomato, which label the presynaptic terminals, in the soma you don't see too much signal enhanced. That is not surprising because there's no presynaptic terminal there, there's no transmitter release at least in this neuron. But in the neurite, in the parts that lights up by the calcium signals, we can see the prominent signal enhancement, and that is due to the calcium activate the transmitter release, showing here. So in the same neurons, the color combination will allow us to look at two different signals at the same time and look at their interactions and separations. How about in different neurons? Remember that at the beginning of my talk, I think that is more important if one could imagine the synaptic transmission, that is the transmitter release from a presynaptic neuron and the corresponding activation of the postsynaptic cells. And here is showing the examples that where we could look at the presynaptic neuron marked in red and the postsynaptic neuron marked in green by the GCAN signal. If you enlarge the signal, you can see that there is a good corresponding of the presynaptic terminal in a way the postsynaptic cell, even touching the postsynaptic spine. And these signals is dynamic. So if you stimulate the presynaptic neuron, which is marked in red, and then look at the postsynaptic signal in the green, what will happen? Well, in this case, if you stimulate presynaptic terminal, you can see the transmitter release from the synaptic boutons, which is shown on the left of that panel. And the corresponding signal, if it reach the threshold, that it will propagate, as one can see that the enhance of the calcium signal throughout the postsynaptic neurons. And this signal can be blocked by the NBQX, which is a specific AMPA-like receptor blockers. So if you apply that to the cells, what you can see, only seeing the local calcium signal originally from the NMDA-like receptor, because if you apply to the cocktail of NBQX and AP5, this signal is completely gone. Okay, what about the other application? Well, this demonstrates in principle that one could visualize the synaptic transmission all by optical means. That is the presynaptic transmitter release from the red and the postsynaptic activation readout from the green. We envision that this could be very useful in the in vivo condition where the circuitry is even more complicated. And the best thing is because of this is an optical reporter, and it can easily couple with the optogenetic approach Dr. Zhou Zhuan was mentioning about, that is the channel rhodopsin. So because we all know that the channel rhodopsin that can by directly absorbing light, that it will allow the cell to depolarize. So because of the availability of different flavors of channel rhodopsin, largely done by the work of Dr. Zhang Feng and Ed Boyden, we could use the same color of line to activate channel rhodopsin, but also use that same light to excite the optical probe and read out the signal in a different wavelength. Then by combining the channel rhodopsin with different flavors of optical readout, one could, in principle, achieve a platform all by optical stimulation and recording. One could achieve to control the neuronal activity and read out whether it's being activated or inhibited inside the nervous system, especially in vivo. And this is a proof principle of experiment showing in C as you can see, if one could couple the channel diopsin tube with the green sensor, then if one use the direct field stimulation, the electrical stimulation, you can see the corresponding increase of the signal reflecting the transmitter release and is subsequent internalization of the transmitter transport. But if one bypass the electrode just using light, one can achieve the same thing. And importantly, this optical signal can be blocked entirely by application of TTX, a specific voltage-gated sodium channel inhibitor. This demonstrates that the light-induced stimulation is not due to the the direct activation of channel diopsin, which is known to be calcium permeable. And we know that the calcium can trigger transmitter release. But the fact that TTX can completely block it, indicating that this transmitter release requires the generation of action potential. And what the channel diopsin does is simply to depolarize the cell to reach that threshold. And similarly, one can do that in an entirely orthogonal color platform that is using a different flavor of channel diopsin, VCHR1, and couple it with the pHTomato based probe I was just talking about. And these two optical spectra is separated in a way that one will not affect each other. Therefore, in principle, one could use two different color of line to both stimulate and record the neuronal activity, that is the transmitter release from the nervous system. And I think that is very useful, especially for in vivo application. Let me just digress a little bit, talking about how we envision that we could use this in an in vivo system by showing some of our work in the fly system. As we note that the fly olfactory system is composed of the several different layers of input and integration from peripheral side to the center. In the peripheral side, the odor receptive neurons, just like the human nose, that will receive different odor inputs that will project into the antenna lobe into about 50 glomeruli. And this olfactory inputs will get further related to the higher brain center, including the mushroom body and the lateral horn, by different projections. One is by the excitatory projections from this so-called EPM projects through the IACT tract to both inner way, lateral horn, and mushroom body. So the same neurons send a carbon copy of signal to mushroom body, similar to humans' hippocampus that is important for linear memory, and lateral horn will be analogous for the amygdala, thought to be important for the innate behavior. But besides these excitatory projections, there is also this different projections that's called MACT, that is sending out from the projection neurons sitting in the ventral side of the antenna lobe. Is different from this IACT. It directly goes into the lateral horn, completely bypass the mushroom body. So this is really interesting. If one look at the lateral horn, that it will receive two different inputs, both from the antenna lobe, but one has a branching out in the mushroom body, and the other one is completely bypassing that. So what is the function of MACT? When I was a postdoc in collaboration with Li Qinluo's lab at Stanford, along with his grad student Liang Liang, what we did was trying to use ways, including RNAi and surgical ways, to remove the MACT, trying to dissect the function of MACT in the olfactory communication. And what we found was that using the advanced two-photon photoablation experiment, we use the two-photon microscope to image the lateral horn neurons, and then looking at its calcium signal, the postsynaptic response, and the control side of the neurons we can also image. after imaging of these two, we then turn up the 2-photon laser power 100-fold, and using that 2-photon infrared laser to cut specifically the MACT track. And then imaging in the same place what happened at the electron hall in the photo ablation site as well as on the control side to see what is the function of MACT contribute to the computation of the electron hall postsynaptic neuron. And what we found interestingly is if we ablate the MACT, we see that there's a postsynaptic lateral hall neuron, the so-called VLPR neurons, has the enhanced response. As you can see here, this is the before the photoablation experiment, and after the photoablation, you can see that significant enhancement of the signal in this part of the lateral hall. In the control side, where the photoablation did not occur, there's not too much difference, demonstrating the specificity of this assay. And we can quantify that it is similar for many different types of neurons. But this response is very specific. If you, what I showed you previously, if you use the food odor and if you cut off the MACT, you see the enhancement of this, the response. But if you use the sex hormone and using that to do the same assay, what you found essentially before and after cutting the MACT, there's no difference of the postsynaptic response. And neural anatomy are showing that this MACT tract contains the GABA, so it's an inhibitory neurons that will regulate this process. So the specificity, as you can see across a different group, of this odor response indicating that the inhibitory neurons has a specific role to inhibit the full related odors, but bypass the sex hormone in the Drosophila. And so the specificity of this odor perception is like what the Chinese have mentioned many years ago. In more than 2,000 years ago, Confucius said, 食色性也 in Chinese. In English is food and sex, human natures. So we have the distinguished response for the Drosophila to their different odors, either to food or to the hormone. So for humans, we also have these different perceptions. This gives us the analogy, or maybe there's an evolution. It's a really evolution conserved in a way that how the brain interprets different odor signals. So what my lab is trying to pursue using our new generation of photo indicators is trying to look at this neural anatomy I was talking about, the MACT here and the IACT into the lateral heart. So how does the specific inhibition shape this odor response to distinguish odors? Well, this inhibition, we know that it occurs in lateral heart. In principle, it can happen in two ways. One is the way that the GABA release directly modulates the EPN neuron in the presynaptic terminal, so the transmitter release get reduced. An alternative way is that the GABA could be act on the postsynaptic side to inhibit the integration of the postsynaptic neuron by the GABA receptors, or both. So one way to distinguish that, we don't have Dr. Zhou Zhuan's nice method for carbon fiber recording from the specific neurons, but we envision that we can put the indicators in the presynaptic terminal and the postsynaptic terminal from the cells at the same time. If the presynaptic inhibition occurs, then we will imagine that the EPN neuron, the transmitter release, will be inhibited. But if only the postsynaptic inhibition occurs, then we will see that the presynaptic release are intact and the postsynaptic inhibition will dramatically reduce. So we have already generated transgenic flies using the pHTomato and similar proteins in a fly olfactory system. As you can see that we are able to use the odor stimuli to trigger the transmitter release detected by the optical signal. So we are in the process trying to cross this fly with the GCAN fly, which label the postsynaptic neuron in green, to look at the regulation of this inhibition at different locus of the neurons. And we think that the same strategy can be applied for many different kinds of neurons because of the inhibition occurs widely inside the brain and in different types of neurons. And finally, I want to spend 2 minutes to talk about the things that are going on in my lab. As Sidney Brand says, that the progress in science depends on many things, but he said that probably depends on new techniques, new discoveries, and new ideas. Importantly, the technique probably comes first. So one of the important goals of my lab is trying to developing better sensors for the neuroscientists, not just my lab, for imaging different parts of the brain and different activities. So we are trying to develop more sensors, not the calcium indicators, because there's already dedicated teams from Genetic Farm and others to develop it. We are more focused on the second messenger neuromodulator release and activation of neuromodulator receptors. And we hope that using these sensors that will enable us to look at the things that none of the neuroscientists has able to think before, and to illustrate a novel principle of the neural development and circuitry forming. And as one example, during our lab's research, we found there's a interesting sensor is also pH sensitive, but in comparison with the traditional sensor, it has a huge stock shift. That is the excitation wavelength and the emission wavelengths has 140 nanometer separation, while traditional EGIP and M-Cherry only has 10 to 15 nanometers. And we caught this fluorescent protein flamingo because of its color and its the pH dependency. In this image, as you can see, one can use the same blue line to simultaneously detect the flamingo red color and the green color due to this same excitation spectrum for flamingo with EGIP and also the large stock shift. So the color separation will allow one to simultaneously detect both. And we think that this will be useful for the in vivo imaging and multiplex imaging for example, with other calcium indicators. And one of the grad students in my lab, Fang Miao, has recently embarked on developing new generation flamingos. Through her screen, she found that mutations that will have a weird pH dependency curve that has a higher cooperativity between pH 5 to pH 8. Therefore, changing the intensity from pH 5 to 8, the signal is increased from the first generation 10-fold to 15-fold. And we hope by our efforts, by direct evolution and selection, we can find more and more sensors, and then we can apply for many more neuroscience questions, not only just in our lab. And finally, I want to thank my postdoctoral advisor, Richard Chen at Stanford, and our collaborator at the Chen Lab. And this is the support I got from Beida, and this is part of my lab, which is just about 1 year old. Thank you.
Questions for Dr. Lee?
See, my talk is so wonderful. There's no questions.
Can you tell how your first application for? Do you say you have the green, which is can combine with the flamingo, and you also have tomato. So is that the three will come together, or you only use two? And what is the advantage with the flamingo compared to tomato? So.
Okay, so in principle, we can just use two if one want to look at the presynaptic neuron and the postsynaptic neurons, which is actually not that many people in the field are using it, mainly limited by the available probes. The advantage of the flamingo, as I mentioned briefly but not in detail, is because of it emit in the red fluorescence, so it will avoid a lot of autofluorescence generally in the relative UV to blue range. So in principle, you will have a better signal-to-noise ratio, especially for in vivo application. And if you look at the spectrum of the flamingo and the EGIP and pHTomato I mentioned, in fact, these three can be both applied at the same time. The EGIP and the flamingo has a similar excitation peak, so it can be separated by the emission peak. But pHTomato has a distinguished excitation and emission peak from EGIP and flamingo, so it can also be separate in both, so three color is possible.
Is that only now for Drosophila? Can that already used for mice? In your
Yes, of course, they are all being genetically encoded. It's just we are really slow on making the transgenic mice and packing the virus. So we start something earlier and fly grow faster, and also we have some interesting biological question that we can be asking to demonstrate proof principle application of these probes.
So haven't they been used in mice yet?
Not yet.
Not yet.
Other questions? Could you comment on the temporal resolution of these indicators?
So this is really a good point. So the temporal resolution of this indicator is not so great. The main limitation, actually, if you look at here, the main limitation is the endocytosis process for this probe are relatively slow. So therefore the re-limiting step, if you want to imaging things are faster, it will have constraints. But the advantage of this probe is because the optical probe can enable the parallel imaging. So one way one can overcome this difficulty is by using the optical indicators to look at in parallel way the neuronal communication, but combining with the electrophysiology, such as illustrated by Dr. Zhou Zhuan, with superior temporal resolution. So then one can balance these two to achieve the best understanding of the neuronal communication.
And that endocytosis, that's just a fundamental limit of the membrane dynamics. That's not something you can engineer by...
So the endocytosis process is known to be temperature dependent. So in the flies, room temperature is relatively slow. In vivo, it will be faster than that, but still is in the range of seconds to tens of seconds.
Yeah, great, thank you. Any other questions? Okay, if there are no more questions, I'd like to thank both Dr. Li and all of the speakers for this session. So thank you again.